So today we're going to be learning about transcription. So last week we were learning about DNA replication, and we learned that DNA has to split between the hydrogen bonds so that I can create two identical DNA strands. Well, that was important in mitosis because I was making daughter cells. But as we begin to move into transcription, transcription begins the process of how we go from DNA to an actual protein. So DNA going to a protein is split into two steps, the first step being transcription. So similar to DNA replication, transcription also begins with DNA unwinding by the breaking of the hydrogen bonds. So as you can see in both images, both the image on the left, the DNA is being split open. We see the same thing here on the image on the right with DNA being split open. Now we know the only way to split DNA is by splitting the hydrogen bonds that hold the base pairs together. We know that DNA only is going to unzip in the area of the gene that I want to be made. So, for example, this particular gene here may be the gene to make the color of your hair. So since that's the only gene I need, my DNA will split open here, and I'm only going to code one side of the DNA. So as you can see in both images, only one side is being copied, not both. So the reason for this is because I begin to introduce a new molecule, which is my RNA. Now we know that RNA is single-stranded. So when I look at it, it makes sense that RNA can only copy one side of my DNA. So in the second step here, we see that the enzymes will match up the free RNA nucleotides to the unzipped nitrogen bases of the gene, which is coming from my DNA, forming a single strand of mRNA, M being the messenger. Now the reason why they refer to this as the messenger is because DNA is unable to leave the nucleus. But since DNA holds all of our genetic information, the body needed to create a way that would allow DNA to send that information out of the nucleus. So the way that our body came up with it was by creating mRNA, M meaning the messenger. So the mRNA will copy the gene from the DNA and then leave the nucleus to go to the ribosome that's going to then code the mRNA. So we see here in step 3 that the strand of mRNA will then detach. Detach means to come off from the gene or the DNA. So this mRNA sequence that's being coded in both images will detach or break off from the DNA and then leave. So you can see here in the image to the right, what I wanted you to focus on is that this process of transcription is occurring inside the nucleus, as you can see. So this purple image here that you're seeing is actually the nucleus. So transcription is happening inside the nucleus, and when the mRNA has coded one side of the DNA, that mRNA will then leave. And it's going to leave through one of these pores that you see here in the nucleus. So I need to come out of the nucleus and into the cytoplasm area where I'm going to begin step 2. So as mRNA detaches here, as you can see, the DNA part that was unzipped because we broke the hydrogen bonds will now zip back up. Okay, we are moving into the second step of protein synthesis. So the first step was transcription. Transcription happened inside the nucleus, and it was when our DNA was being copied into messenger RNA. And we know that right at the very end of transcription, that messenger RNA was leaving the nucleus and into the cytoplasm. Now, the reason why the mRNA wanted to leave the nucleus and into the cytoplasm was because the messenger RNA, as you can see here at the top, needed to find a ribosome, which is floating around in the cytoplasm. Now, as you can see, step 5, which is the first step of translation, is the newly synthesized mRNA strand that just left the nucleus will combine with the rRNA. Now, the R, little r, is standing for ribosome, which you can see in the image to the top. The rRNA reads the mRNA every 3 bases. Now, those 3 bases are considered a codon, and each codon has a particular or specific amino acid that is being called. So as you can see here at the image at the top, the ribosome read AAA, and it coded for lysine, which we see the LYS here. Then the ribosome reads GUG, and we get valine, VAL here. So the ribosome is reading the messenger RNA every 3 bases, and these are considered my codon. Now you can see we have this other molecules here that are bringing in the amino acids, which brings us to our next step, step number 6. In the cytoplasm, there are these molecules called tRNA, the T standing for transfer. Now, you can also remember that this is a tRNA because of the shape of the actual molecules in the shape of a T. On one end of the tRNA molecule is an anticodon that you see here at the top. Now, this anticodon is a set of the 3 bases that's going to bond with the actual codon. So, I don't want you to be confused. Codon is on the messenger RNA because the messenger RNA is the one that has the code that he got from DNA. Anticodon, the word anti meaning the opposite, is going to be the base pair that will bond with the 3 bases that the ribosome is reading from the messenger RNA. So in this instance here, we had AAA, so that would be my codon. UUU would be my anticodon. So the anticodon is basically the base pair to the 3 bases that I am reading at a time. Also, on each tRNA molecule is 1 amino acid. So we know that it takes 3 bases in order to give you 1 amino acid. And each tRNA molecule contains different amino acids, as you can see from the image here. Okay, in this next image here, we're going to be talking about how the tRNA contains the anticodon that must match the codon found on the messenger RNA. Now the codon and the anticodon fit together like 2 pieces in a jigsaw puzzle. So just for instance, if my codon is AUG, then my anticodon would be UAC. So I'm only looking at the base pairs. Since I start with an A goes with U bonds with A, and G bonds with C. Now the process will move along to the next codon. So as you can see here at the top, we have an arrow that's moving in the left to right direction, meaning that the ribosome is moving from this spot to the next one to the next one and continues down all the way to the end. So as the ribosome continues to move, more TNA molecules are needed to bond with the new codon. So here we see that we have UGC, and we see this tRNA molecule over here is about to come and bond with the next codon. So they're getting ready to bond together. So you can see that the amino acid brought by the first tRNA bonds to the amino acid brought by the second. So what this process is talking about is this process right here. So we see that we have our growing amino acid chain here. So our growing amino acid chain is about to be transferred onto the new tRNA since ribosome is moving left to right. So you see we have this E slot right here, E for exit. So as the ribosome moves down to the next one, I need to kick out the tRNA to make room for the new codon. So this tRNA here will move over to the exit site and then leave, and the entire amino acid chain is going to be transferred. Then I move on, I'll transfer it onto the cytosine, transfer my chain onto my tyrosine, and I will continue the process with each tRNA molecule coming in with 1 amino acid. I transfer it, and then I exit. And my tRNA here is empty, meaning that it has no amino acid. So steps 8 and 9 will continue, each new one coming in, transferring it. So because the tRNA is transferring these amino acids, that's where the T in tRNA is coming from, because of the process that it is doing. Now, we know that amino acids are a monomer, and the monomer that they are creating is a protein. So the final product that I'm getting from translation is a completed protein product. And these protein products are going to be created by the amino acids that are being brought based off the codon that the messenger RNA is bringing me. So I just wanted to give you an overview of both processes together. So we see we have my nucleus here, and in my nucleus I have my DNA, and my DNA is being split. As my DNA is being split, I see that I'm only copying one side. So this one copy that I'm making here is my messenger RNA. After my messenger RNA has copied the gene, the messenger RNA will detach and leave the nucleus. Now I need to leave the nucleus and enter the cytoplasm so that I can begin the second process, which was translation. So I need to translate nitrogen bases into amino acids. So since it's two different languages, I'm translating nitrogen bases into amino acids. So that's where the process of translation came from. So you can see that I'm reading my messenger RNA every 3 bases. And we know that my messenger RNA is the codon. I see my tRNA molecules coming in, and I see that each new tRNA molecule is bringing me 1 amino acid. So it takes 3 nitrogen bases to give me 1 amino acid. So as they will bond in here, the growing chain of amino acids that you see here will then be transferred onto the next one, and then this tRNA here that's holding the entire chain will leave to make room for the new tRNA molecules to come in. Last thing was I wanted to be able to compare and contrast DNA replication and transcription, particularly using these images, because most of the times when you see your state exam, they're going to try and give you images of either one and will ask you what process is being shown. So in DNA replication, I see that I'm going from 1 DNA molecule to 2. So I see that I'm coding not only one side, but both sides are being coded. So when I look at this, I should see 1, 2, 3, 4 strands of DNA, since I'm making 2 DNA strands. Now when I look at transcription, I can immediately see that I'm only coding one side. So remember, transcription splits the DNA and is only coding one side, not both. So when I'm coding both, I'm replicating. When I only code one, I'm in transcription. The other thing is if I look throughout all of these base pairs, I don't see any U. Every time I have an A, I see a T because I'm making more DNA. Now in the process of transcription, I'm not making more DNA, but I'm making an RNA. So you can see here in this one that's being coded here, that every time you have an A, instead of a T popping up, I see a U. So this is another signal to me that I'm looking at transcription and not replication, since transcription involves RNA, and I see U bases here, so I know that I'm dealing with transcription, not replication, because replication will only use a T since you're making more DNA, but transcription, you will see T's and U's because DNA is being coded by RNA.